An overview to biological specimen processing using histological techniques. The Mise way! Hi everyone, I'm Ayaka.
And I'm Jam.
And today we're going to be talking about histology. So from Sensei's room right here, we're going to show you first how to get there. Sensei's room is here in 353. Then you want to go this way, past the molecular room, 354. This is the histology room, 306.
San Maru. What is 6?
Roku. So to unlock this room, you go here. So once you first come into the room, you put the key and lock there. And when you're done at the end of the day, make sure to take it and lock it up again so that other people can't come in. So now we're in the histology room.
Yeah!
So let's take a look around.
Take a look around.
Yeah. Oh my gosh, look who we found in the histology room. It's Kairi-san, Emmeline.
Sumimasen!
We're here to learn about histology.
Oh, you come to the right place.
Oh. We're very excited to learn.
So, histology is defined as the anatomical study of microscopic structure of animals and plant tissues. So today we will give you an overview on how to process the biological specimens using histological techniques based on Emmeline's study on soft coral reproduction.
So what we'll be needing today is a scalpel, pencil, cassette, one of two types, pink or blue, petri dish, two tweezers, one with the red formalin tape, one without, and your samples. Don't forget your 70% ethanol. The purpose of this step is to select the proper areas for examination. For reproductive studies of soft corals, the target area are the gonads, so we have to make sure that we capture the proper tissue orientation. So after we've taken out the sample, we have to correctly cut it so that it is in that proper orientation, and then we can take that sample and put it in the cassette. Once that sample is in the cassette, we can then take the entire cassette with the sample in it and put it in your jar of 70% ethanol. And finally, we can take what remains of the samples and put it away. For this next part, there are two types of ways that we can process the tissue: manual, using hand processing, and automated, which is using a tissue processing machine. For our demonstration purposes, we will use the automated way using this Ayaka ASP200S, which is a modern enclosed tissue processor. It's really efficient to use the automated way, especially if you're processing a lot of samples for reproductive studies. This part of the machine contains the paraffin, but it won't open because it is solidified. The other side will open, and it is where we will put the cassette and samples. To turn on the machine, simply push this button up. Here we can see that we can open this side up, and we will show that the inside paraffin is now liquid. And this is the solidified paraffin. Whenever you use the machine, don't forget to record your usage in the lab book. So let's get started with this process using our samples. So first, make sure to select the right settings, and then you can kind of look at all of the reagents in there, which is important for dehydration, clearing, and wax infiltration, to ready for paraffin embedding in the next step. And we can then start to put all of the cassettes in once the correct settings have been selected, and close it up. Go back in. Make sure everything is set right and input the number of samples that you've put in the machine, and we can start the process. We can then take a look at how much time we have left at each step.
8 hours later.
So now the first 8 hours is finished, and we just have to wait for the retort, which is the area that you put the samples in, to empty. Once the retort is empty, you can select OK and remove your samples. Once you've taken your samples, make sure to clean up the machine so we can start to finish this part up. Once you finish cleaning the machine, we'll start up the cleanup setting, where we will select the standard program, press OK, and it'll start.
A few moments later.
Once the program ends, make sure to click OK with the pop-ups, and we're ready to finish off. Once the cleanup program ends, don't forget to wipe this part of the machine off with ethanol before closing it up. Finally, once that's done, we can start to close up the applications and power down the machine. So the next part is embedding, and first we'll show you how the machine is set up. So this is the embedding machine. Press that button to turn it on, and the top compartment has solidified paraffin that will melt. That also has paraffin. Pushing this button will turn on the left side of the machine, which will keep the samples cool. These buttons correspond to the different parts. The left one corresponds to this paraffin container, which is currently solidified. The next button corresponds to the top part, which has the paraffin in it. The next button corresponds to this small area here, which is kept warm, and that last button shows the other paraffin container. And here you can see the temperature that it is heated to. Next, we can start to prepare our samples. So first we want to open the cassette where we have our sample. We'll use the tweezers to take the sample and put it in this mold. We will then put a little bit of paraffin, melted paraffin, in it, just enough to cover the bottom, and put it in the heated area so you can still move it around and the paraffin remains melted before transferring it to this little spot that cools it down. And we'll wait for the paraffin to solidify slightly before adding some more in. After filling this with paraffin, we will put the top of the cassette back on and wait for it to solidify slightly. Once the paraffin is solidified slightly, we'll add a little bit more again on top of that cassette lid. Then let it cool a little bit again on this small cooling pad. And then add a little tiny bit more paraffin in the end, and transferring the whole thing over to this cool area. Once it's solidified fully, we can sort of help nudge and scoop the sample out. and it'll look something like this, and we can just put it back onto the cooling pad. Once we have our solid samples, we can then start to cut into it a little and just create a sort of thinner box around the tissue so that we can put it in the microtome. And again, don't forget to log your usage after each machine. We'll then move on to this next part and first show you the microtome. First, we can adjust this screw to put the sample we just made in it. By turning the handle on the right, it kind of moves the sample up and down. By adjusting the small lever on the left, it kind of brings the sample closer to or farther away from the blade. By moving this lever, we can also kind of adjust and tilt the blade. We can also unscrew this part of the microtome, and that allows us to take out the blade to clean it and also clean the surrounding areas. Next to the microtome, we will also need a water bath that we can adjust the temperature for to put the cut samples in. And finally, we'll also need a slide warmer, which we can put the mounted samples on to allow them to dry. This is the whole process of what cutting the tissues looks like. This was my first time, so take it from that perspective, but you can use the lever to kind of cut the samples on the blade like so. And once you're done with that, you can use some brushes to kind of elongate the portion that has been cut and put that into the water bath. This is Kairi-san, who's also showing us how to do it, especially once you get more used to it and can do it faster. Next, to mount your samples, you want to write down your sample name on the slide. And with a lot of finesse and delicate work, we will try and put that sample onto the slide before transferring it onto the slide warmer. Now there's also another way to do this whole microtome process, beginning with readying your slide, writing down the names, and adding millicue water on top, which can allow you to make sure that the water is free of any potential contaminants. So once you've prepared the slide, Emmeline-san will show us again that you have to cut the sample and then transfer that directly onto the slide. And then move the slide with the samples on it directly to the slide warmer, and we will begin to start removing some of that water, making sure that the sample is in the right position and letting it dry. These glass slides then remained on the slide warmer for around 24 hours at 37 degrees Celsius, and staining can be performed after this. Once the slides are dry, put it in this rack and we can get ready for staining. Don't forget to write in the notebook after using the microtome as well. So now that we have prepared the slides, we can move on to staining. These are all of the reagents that we are going to need for this process. In addition to the reagents, we'll need our samples, slide covers, and the mounting reagent, which is optional but can help preserve your samples longer, and the dropper to put the mounting reagent on. So once the slides are dry, we can take them over to the reagents, and you'll put the entire thing into each reagent for a specified time. The order of how to stain is quite long, and so we'll put a table here with detailed instructions on which reagent for how long, so please pause the video to look at them. So now, once you've gone through all of the reagents and maybe used the mounting reagent, you will have stained slides that look like this. And we can put it under the microscope to see what the final results of the tissues look like. If you don't know how to use a microscope, please refer to the microscope video, but here are the final products of some of the tissues. Super cool! And thank you so much for tuning in today.